Hello, good morning to everyone. This is another Meet the Scientist session. We are here today with two scientists from BIFI in Spain, in Zaragoza, and Socientais. It's Eduardo, and I don't know the name of your colleague, but you will introduce yourself now in a minute. We will talk today about digital science, citizen science. And if you have any question, you can make it in the chat or you can even enter in the session. So hi, you can start whenever you want.
Well, good morning. My name is Eduardo. I'm, as Enrique says, thank you for the introduction. I'm a researcher from the Institute for Biocomputación and Physics of Complex Systems from the University of Zaragoza. I'm a computer scientist. The institute is a multidisciplinary institute for biocomputation physics. So most of us are or biologists or computer scientists or physics. So, well, my research line, it's mainly citizen science, but first of all, we present the institute. It's one of the most relevant e-science centers in Spain. Both as resources providers and scientific end users. We have a whole infrastructure of with several technologies for high performance computing, clusters, memory, and all about biology that Cote, also known as Jose, will present later. In computation, the main research lines that we have is high performance computing, grid and cloud computing, citizen science, and some space purpose computers. The research in HPC, high performance computing, it's done through several national and European level projects that try to study the new trends and technologies around supercomputing. Actually, now we are in a project. A European project funded by the FP7 framework with China, with the Tianjin supercomputer, that is one on the top of the top 50 supercomputers in the world. So the main tasks are the operation and support for those supercomputing resources that are hosted here at BIFI. We have a data center, and yeah, the idea is that they have a wide experience in the management of that infrastructure, and they try to give support not only for the researchers in BIFI, but also researchers out of BIFI. Anyone that wants to carry out some calculations or some projects, they can put in contact with us. And the idea is to try to give support to everyone that needs to run scientific computations. So we provide.
So, wait a minute. So anyone can do. So for example, me, I could go to you and you could lend me your supercomputers.
We'll have to negotiate about that. But yeah, the idea is that we provide support to researchers around the world. So
and I can run anything there in those computers, anything related to science. I mean, or do you have specific programs or specific languages for those computers?
We'll need to port and usually we have several computers that run several operating systems, and the idea is we'll need to port your programs to our platforms. So it's possible, but in the middle from your application and to be able to run that application in our infrastructure, we'll need to do a porting. But actually, the people working on that are also experts, and they have a big background. I mean, they have been working on that for years, so they are experts on how to port applications to the infrastructure.
Okay.
Yeah. So actually, the idea of the center and the HPC and the grid and cloud computing and mainly citizen science is to provide support to other researchers, so they can use our infrastructure to run their own projects. Okay.
Okay, perfect.
Yeah, by the way, if anyone wants to have any question or whatever, just write in the chat, and feel free to interrupt me at any moment. Okay. And well, yes, as I was saying, some of the services that they provide are also porting, optimization, and parallelization of applications. The grid and cloud computing team usually work also in national and international projects related to distributed computing, either cloud or grid computing, and integration of those technologies, not only research but also companies, since there are also companies that can port their applications to run on mainly on the cloud that they are setting up now. Also, well, as you know, computers are pretty inside our lives now, and for some kind of specific operations, mainly in science, still computers are not as good as it should be. I mean, they will need a lot of calculation, a lot of time to run several calculations. So we also design and build some machines, some computers that are for special purposes. So for some specific problems, conventional machines that are not enough are becoming a new machine that can develop and perform any concrete heavy calculations. And that's another part. And in that time, in 2008, the first one of those computers is called Janus was built, and in July 2015, the Janus 2 was the Janus 2 was built, and also it's mainly for that kinds of heavy calculations that right now are running some algorithms for spin glasses. That is a kind of another line of the physics researchers on BIFI. So, and the last computer science line that we are working on is citizen science, but we'll talk thoroughly about that later. So I will skip this part now.
Okay.
On physics, the four main research lines are spin glasses, molecular dynamics and electronic structures, physical modeling of biomolecules, and complex networks. The later one, the fundamental idea is to discover the structure of interactions between the components of the systems and the emergent behavior of many body systems coupled to the underlying structure. The ultimate goal is to understand the general principles governing these biological, social, and technological systems in order to be able to predict, design, and control the behavior of a wide range of real systems. So some of the lanes that they're working on are communication networks, like social networks, epidemiology, evolutionary biology, network theory, multi-scale diseases, online social systems, socioeconomic systems, or some kinds of systems biology. So that's the physics and computation parts, the research lines we're working on. The other part of the institute is for biology. I think Jose has much more to say than me, so I think he can introduce himself and some of the links.
Hi, how are you?
Hi.
Well, my name is Jose, but I am also known as Cote around here. That comes from Jose. So I am a researcher and a professor here at the university in the bio part of the BIFI. We have different lines here, and major research lines at BIFI are related to protein structure, protein folding, protein docking, interactions between different types of molecules, usually biomolecules, proteins, protein DNA, nucleic acids among themselves. We have also a line that's devoted to drug delivery studies. My own line is called stem cells and apoptosis, so we're also studying cell death and stem cells differentiation, for instance, and possible applications in cell therapy. And perhaps one of the major lines that differentiate us from other centers is deals with chemical screening, basically screening of chemical collections, looking for compounds that can become drugs for different diseases. And that's actually where this research in social science, this research line with cell spotting fits here. Another line also deals with oxidoreductase, basically enzymes that work with electron transfer, mainly during respiration or photosynthesis. That's basically a summary of the different lines in the bio part of the BIFI. So if you probably want to know about what's behind this cell spotting line, basically what we were doing was we were looking for chemicals originally. Citizen science, yes, but you were talking about that.
Yes. I think we can talk about the experiment later.
Yes.
So okay. We introduce the rest of the institute. So.
Okay.
So in this link, you can see the institute, or we have around 20 laboratories and 22 offices distributed in three floors. And here you can take a look to the supercomputing lab that now Fran, the third researcher here today with us, represents parts of the cluster. So in order to access the cluster, that I'm going to provide you a URL that is this one. They will ask for username and password. So the username is global. And,
no, the username was Excursion, right?
No, username is global, and the password is Excursion.
Sure, I did the opposite.
And you could access?
Yes, the username is Excursion and the password is global. I'm watching the camera.
So there you have it.
So I wrote it in the chat for everybody. So in the username you write Excursion, and in the password you write global. These are people to access and to see the live webcam that they can control. This is their data center. So this is how they monitor their... Okay, if anyone has any problem, just please ask in the chat. Okay, you can continue. I think we see it, yeah?
Yeah, I will show some different kind of supercomputer here that we have. We are trying to cover different kind of computation. So I can show in the, if you click, I don't know if the people have to click, or if I click, or all the people say the...
you come to control the camera and they just see what you do.
Okay. So here is not Memento, but in the left of this computer, I can do in this way. Sorry. Okay, here is Memento. Is a kind of computation. Here we have a lot of memory for the computation. Each node of the supercomputer have 200 gigabytes, more or less, of memory. So we can do a kind of computation that needs a lot of memory for each calculus. We can see another important computer is Janus. Okay.
This is the one that Eduardo told.
Says before, yeah. This, we are doing a very special kind of computation here, because each processor only can do one kind of computation. This is not like our computers that you can do a lot of different things, but here we can do only one kind of computation, spin glasses computation. But this computer do that very, very quickly. Yeah, and maybe important, you can show. Here we can see the diversity is host, but I can talk a bit about IVRCs. In this kind of computation, we are doing the calculus in the computer of the people, not in this computer. So we are sending different programs to the people, and the people return the result to our servers.
How much is one of these machines, big supercomputers?
How much?
How much does they cost?
Because it depends but I don't know how to say each node. For example, each node of one of these computers, each node is one of the lights of this. So each node costs about 8,000 euros, more or less.
So it's not so expensive, yeah?
No, it's not. But sorry, 800.
Yeah, 800.
Yeah. And this, the Memento ones, for example, about 400,000 of euros.
Yeah, that's a lot of money.
That's a lot of money. Yeah.
Okay.
So more or less, a brief resume is that we are trying to cover a lot of some different kind of computation, and we need some different kind of computers to do that. Yeah.
Okay. Thank you, Fran. So,
and one last question. What do you use this webcam for? Just to monitor, to see that the machines are working, that they are up, or just to see if anyone can be there?
So the point is that we can see that everything is working fine, because sometimes some things can fail, and we can check if the lights are fine. And we can see also if everybody is here. That don't have to be there.
Okay.
Okay. Thank you, Fran. Yes, as you can hear, actually, these data centers cost a lot of money. So that's why we thought about another way to do science. And thanks for moving to the presentation. So the idea behind citizen science, that is what we do. Well, this is the structure of the presentation. First, we talk about citizen science, about the projects, and finally about the experiments, focusing on the cells spotting experiment. That is the one that the driving force is Jose. So what we think is that everyone can do science. So in such a way, researchers have the help of the general public and in some specific activities. So we can save a lot of resources that we can afford in this moment. So citizen science is based on scientific activities in which citizens participate in things like data collection, analysis, and dissemination. It's voluntary, conscious, and informed. So citizens are keen to participate, and then they can do it in two ways: or in active way, where citizens use their intellectual skills, resources, and time, or in a passive way, where they only provide some resources, some technological resources. So to sum up, citizen scientists are those who dedicate their intellectual skills and/or their technological resources to find results with social utility. The advantage for the citizen scientists is the acquisition of new skills and knowledge on the field that they are working. Of course, they develop critical thinking, since they have to do some kind of analysis, not only when we talk about qualitative thinking, where they actually participate in the kind of analysis, they have to think about what they are doing. So they develop some type of critical thinking. They do real research. They participate actively in research projects. Of course, since they are participating in such projects, they participate in science. So they acquire a real and accurate perception of what science is. And the most important is that they contribute significantly to the advance on science and technology. That's why we say that citizen science is the link between science and society. On the other hand, of course, for the researcher, he has a huge crowd working for him. So volunteers can help him to generate useful data that will be much more costly and time-consuming to obtain without the help of such people. It's just what we think that citizen science means. It's and what the aspects of society that they involve, like democracy. We have to also work on the policies for the European and in general the governments to get involved in this process of science. It means innovation, co-creation, learning, empowerment of people, participation, and mainly openness of science to the general public. So to separate somehow, we have the active participation and the passive participation. The later one is where a user only provides some kind of technological resources. And as this project, SETI at Home, it was developed some years ago in order to find extraterrestrial life, analyzing some kind of sounds on waves. And what volunteers have to do is just to install a little program that receives some calculations to do and give back the result to the researcher. Civis, it's the foundation that we are working on, and it's some kind of platform to perform the projects like the SETI at Home, alike to the SETI at Home. And well, the same Rosetta at Home, it's another kind of project done for protein folding, design, and docking. This is what we call passive participation, but in active participation is where actually the users not only has to provide technological resources, but also they participate actively in the process of science. And there are some of the main projects on the world. Galaxy Zoo maybe is one of the most well-known, where people started analyzing galaxies, images of galaxies, and to creating a map of the whole galaxy. Audubon, it's an association from the United States, and actually they started doing citizen science like 100 years ago, where just the volunteers counted some kind of bird when they are migrating, and they just wrote the letters to the main researcher to say saying how many birds have they count in their place, so they can control the birds migration around the states. Another of the most well-known projects is Foldit, but we'll talk about it later. So behind that two separations between active and passive participation, we can create a more concrete, more fine granulated kind of project that we divide among volunteer computing, experiments, sensing, and thinking. Volunteer computing, as we explained before, is when a user just provide technological resources, but it's not active in other way. For instance, Boink is the platform that is used by SETI at Home, as the project that we mentioned before. Boink is a platform. It's a software that the volunteer install on his computer, and when you can configure, you can set the filters on when Boink will work. But usually when there are some free time on the CPU, the software is launched, and it runs algorithms, it runs calculations of the project that you have selected. So Boink receives some packages of work, and in the free time that the computer has, it runs those algorithms, and it does some calculations. When it finishes, it gets back the results to the researcher. Here you have some of the statistics of Boink. as you can see, the whole Boeing has more than 2,700,000 users. What is a lot of hosts and a lot of computation power that it could be done in other way, or not in a cheap way, at least. So,
just one question. This is doing that with desktop computers, right? Not mobile phones or tablets.
They are developing some projects for mobile phones also, but they are still under development. The most important, the huge range of devices used are desktop computers and FPGAs.
GPU.
Actually, GPUs are pretty powerful and usually run much more calculations, but it's not the things that usually people have at home.
Yeah.
Well, another kind of projects within citizen science are what we call voluntary experiments. That user, let's say that they do science or without knowing that they are doing that. Usually they are doing through some kind of applications or games, like Foldit. It's one of the most well-known. Foldit is an online game developed in the States that on UK, I'm not sure right now. And they present to the user this kind of images of proteins that they have to unfold. The user, it's a game, it's a mess of proteins that they have to unfold and separate the different parts. This is really important because it's a really difficult calculation to do through a computer. It's while for a human brain, it's pretty easy, let's say pretty easy to see how can they just separate, and it has a really significant contribution for protein docking and folding. Also, another paradigm, let's say, it's the voluntary sensing, where user acts as a sensor. Sorry, wait, I need water.
So here the user lends his sensors, right? His mobile phone, his position, his...
Yeah, but not only the sensor. Himself can be a sensor. For instance, here in GripeNet, that is the web page that appears, the image that appears on the left. It's a GripeNet. It's a project to monitor the epidemic, the influenza,
right?
Influenza, yeah, but mainly the epidemic. Spreading. Thank you. So the idea is that every user can weekly fill a questionnaire about if he has some kind of symptoms from the flu, and in that way they can monitor and create maps about how the flu is spreading around the country. This is GripeNet is the one from Spain, but it's a European consortium of 9 or 12 countries. So they can control and monitor how the flu is spreading through Europe. And there, the user is not using, the volunteer is not only using a device to take pictures or something. It's himself is the sensor. But also, like on the right, EpiCollect is a web page where everyone can create their own project, and through the use of a mobile phone application, everyone can share or pictures, videos, or just upload some kind of information that the researcher can need. And finally, the most interesting thing, at least for me, it's volunteer thinking. That are applications where the user is actually pretty active in what they have to do. Maybe the most well-known is Galaxy Zoo. As you can see on the left, they started presenting some images of galaxies that the user has to classify among 3 types of galaxies. And I don't remember exactly the numbers, but I think in the first year they got like 3 million of images classified. but it's really, really a lot. So, and it's pretty easy because only the volunteer doesn't need a really strong background on astronomy. It's just to see they have just to see the images and classify them on the 3 types that you can see on the right part of the Galaxy Zoo screenshot. So if the galaxy seems like the first one on the left, they just click there. So
you also have to categorize to see characteristics and then see similar to.
Yeah, exactly. So it's pretty easy, and everyone can do it, and also they are really contributing. The most interesting part is in Galaxy Zoo, they have a really strong community behind the project, and they started to write the researchers because they found some kind of small red points that they didn't know what they were. And then they started to talk among them and ask the researchers about that. At the end, they discovered a new kind of galactic body. And actually, they published several papers in journals with the discovery that they did. And they were the volunteers, not the researchers who did that. That's the way that we consider.
That's curious. Do you know any experience with this kind of experiments, or volunteer computer, volunteer thinking, volunteer sensing with students? Do you know any teacher can participate with their students?
Yeah, or experiments. As we'll present later. I mean.
Okay. We can keep that for later.
Yeah, but that's. I mean, we strong believe that this kind of science fits pretty well in education because, like the astronomy, for instance, the students can learn a lot about astronomy, or it depends on the project, but they can get easily a lot of background on the field that they are working on, and yeah, I mean, also they are collaborating to science. Another example is the one on the right, that it's crowd crafting. Crowd crafting is a web page that it's working on top of Bibosa, that is the citizen science infrastructure that they use. For instance, this project, it's about lichens to measure somehow the pollution on the cities. So all these pictures are taken through the EpiCollect framework that we mentioned before. These pictures are saved in a server and displayed in the application. So through a really simple classification about the size of the lichen and what kind of lichen it is, that is just select three kinds of samples. They classify the lichens they classify the lichens that are geolocated. So they can create a map about how the pollution is affecting all the trees and the vegetation on the cities. So some other things that we can do with citizen science are like the hackathons that are within the participatory activities. We did the two of this year, and the hackathons are some kind of meetings where researchers, citizens, journalists, whoever wants can join them. At the beginning, we present several ideas, and during 1-2 days, we are working together to develop such ideas. What happened with the presentation? Wait a minute.
What is
I'm having some problem with the presentation. The previous one.
Okay.
Here.
That one. Yeah.
And some other experiments, like for instance, for the prisoner's dilemma, we need to the participation of a lot of people. So we create some kind of, it's moving there.
Yes, the presentation is moving. I don't know if anyone is moving it. Let me set the one.
Thank you. So yeah, there are some kind of experiments where we need the participation of a lot of people. So we create big meetings where at the same time everybody can work online to develop some kind of experiments. So from BIFI, we are coordinating and leading a European project funded by the FP7 program, 7th framework program. That tries to coordinate all agents involved in the citizen science process and mainly set the basis for this new kind of open science paradigm in Europe. We promote the uses of science infrastructures composed of dedicated and external resources, including professional and mainly the amateurs, so the volunteers. The idea is set up a network where infrastructure providers and researchers will recruit volunteers from the general public to perform science at home. And one of the most interesting parts of the project is also we are writing a green paper. The first year was the green paper, and we are working on the white paper for the second year that intends to set the basis of the citizen science in Europe by means of providing policies to the recommendations for policies of the European government and how they can face and take advantage of this new paradigm. So the project tries to increase the participation of the large public to disseminate, to make citizen science well known, and thanks to that, increase the participation of the large public in the process of science. Generate positive contributions to the research, foster the cooperation among researchers and volunteers. Of course, analyze which is the real impact of that paradigm, either in scientists, in volunteers, and in society. So here's what I mentioned about the white book we are writing.
Mm-hmm.
The idea is generate knowledge, and that through that knowledge we can present the paradigm to the EU, the politics, and demonstrate them that this can actually work, and that volunteers are a really powerful tool that can be used in research. So
this book is like an extension of the green paper that you prepare, right?
Yeah, sure, exactly.
And it will be for free, or I mean,
sure. Actually, if I search now the
no, we will send it after to all the participants.
Yeah, actually, yeah, right. Yeah, but the green paper was the first draft that we wrote it for ourselves, but following the philosophy of the project, we opened the discussions. So several dozens of people collaborated and wrote their opinion and help us to write ideas, set up and foster the discussions. And as a result, we got the green paper. And now we presented this in an ICT event in Vilnius for the European program for Horizon 2020. Mm-hmm. And now we are working on the white book. And actually, we open again the discussions. And in 1 month, I think the tool will be available on the web, so everyone can contribute with their opinion and talk and discussions and so on.
Okay.
Well, this is the partners and so on. Sorry, it's a kind of corporative presentation on this part. This is the website. You can take a look if you want, socientize.eu. There we have the links to all of the experiments that are active now, and a lot of information about citizen science, other projects. Actually, we have a section only for citizen science projects where teachers can take a look. So maybe not only our experiments they can find interesting, but they can take a look. So they have a lot of resources that maybe it may be useful for you. And well, this is the experiments that we are working. The three on top are the ones that are active, while the three below are under development yet. Under the development yet. Antimatter is an application that we're working with CERN, so it will be available hopefully in 2 months, more or less, and it's about antimatter. Urban Bees is a kind of participatory project where we can, the idea is to evaluate how pollution and the cities are affecting the urban bees, are affecting the bees. That can give us an idea about how are we affecting to all the environment through the pollution and all the expansion of the cities and so on. Mm. The third one, saving energy at home. It's about energy efficiency at homes. So the idea is similar to the GripNet. Users has to give an input about their habits at home and the consumptions and the energy that they have. And the goal is twofold. On one hand, the researchers get a lot of information about the behavior of users at home, so they can work on the theoretical models to improve the energy efficiency and energy consumption at home. And also users get feedback from how their energy consumption at home it is actually. They get tips on how to improve the energy consumption, customized tips according to the information of the house and their own habits. So the idea is that they can improve their energy consumption at home. Well, among the actives, the first one is the MinPaths. It's a project on linguistics. It's a volunteer experiment, as we said before, where it actually is a game where we have a source and a target. A word that is the source and a word that is the target, and the words related to the first one. So you have to try to achieve to reach the target through the source. You have an excursion. Wait.
I have seen any game like that.
Yeah, actually, it's pretty funny.
Yeah.
Addictive. So you have the source that in this case is experiment, and you have to get bond. So you have all the words related to experiment, and when you select one of them, the selected word becomes the source, and you in such a way you are building the path to get the second one. And the results are really significant for web semantics, web mining, text mining, and in the business world, some kind of applications are really based on semantic analysis. So this is
do you consider the user profile when doing this kind of experiment? I mean, depends on a pupil, an old person, a young person.
No, because the idea behind the research is that everyone, independently to their background, their life, has the same in their mind. They really have a pretty similar path to get.
Is that right? So how do you confirm that hypothesis?
Not yet. We are working on that. As soon as we have the results.
Because I think maybe it can vary even from country to country.
Yeah. Actually, it depends more on the language. Actually, it's English-speaking application, and the difference, what can create a difference is if you are native or not. Because the links that you create among the words depends on your linguistic background. But the idea is that it depends on your linguistic background and not in your own background, in your life.
Yeah. Oh, okay, that's curious.
Yeah, and yeah, it's really funny. Later we will share all the links. So sorry, you can take a look and have a try and more than one because it's really funny. So
yes, indeed, for everybody, in the end, we are recording it and we will send the presentation, the links, also a couple of videos, very interesting. So you will get a report with everything.
okay, thank you. The second application that is the one that we have active and available now, it's Sun for All, that try to study the phenomena occurring in the surface of the sun and to understand the solar activity and the solar cycle. It's carried out in a research of the University of Coimbra. So they have a lot of observations, a lot of images that have to be analyzed. So the volunteers must count the sunspots and the groups of sunspots that they can find in the images that we are providing to them. So here you have as a screenshot. So the idea is, all these white sunspots must be analyzed, and thanks to that, they can study all that phenomena that is occurring on the surface of the sun. And well, here you have a small screenshot of the saving energy at home that it will be available hopefully in less than 1 month, and you will be able to take a look then.
Okay.
And well, now the last one of the most interesting applications that we have available is cell spotting, and I think first of all I will leave the word to Jose, because he's the driving force and the research, the head researcher behind the experiment. So I think he can explain more better than me.
Okay. Okay. So the beginning of this experiment came from a project where we were looking for chemicals that would induce a specific chemical, specific differentiation in stem cells. The idea is to get any type of cell that we need for cell therapy.
Mm-hmm.
But it happens that stem cells and tumor cells have some features in common. For instance, they are immortal, they can be kept in culture indefinitely, and also they have some similarities in their metabolism with tumor cells and stem cells. So we have selected some compounds in stem cells and we wanted to test them in tumor cells. And that's where the project started. Basically, what we do is that we culture cells in flasks that they have many different wells. And in each well, we add a chemical compound. And then we try to identify which chemicals are going to kill cells and how. Basically, we're interested in a type of cell death that's called apoptosis, because that's physiologically controlled and it's not going to produce inflammation in the organism. Cells are going to be silently eliminated. So basically, if we want to kill a tumor, we don't want to produce an inflammation in the rest of the body. So we're interested in this specific type of cell that's apoptosis. And that has some specific morphological features that can be seen using microscopy. We can label different organelles in different colors. So basically, we know not only what's happening to the whole cell, but also to specific cell organelles. And that's where people at home can help us, giving them some rules and a guide that they can follow to identify different processes in cells. They can analyze thousands of images for us. So basically, that's the summary of this. I don't know if I can move through this now.
Yeah, maybe I can move it for you if you want. Okay.
Yeah, so basically what you have here is a field of microscopy. Nuclei are labeled in blue. These are fluorochromes. Basically, they're going to emit light. When they are illuminated with a certain wavelength, they're going to emit light in another wavelength. So we use this blue compound for nuclei. We use another compound for mitochondria that's green. And basically, we are overlapping 3 different images: blue fluorescence, green fluorescence, and bright field microscopy. That's basically the standard microscopy where we see cells with a gray background. And then the idea is to identify features here. I can probably go through the application. Look, it's over.
we can share now this link.
Wait a second. There is a question in the chat. Is this a desktop application or an Android application?
Yeah, it's a desktop application. It's a web application that you can access through the browser. And
so if it is in the browser, maybe you can access it with the phone, or if not,
it's done with HTML5. It's done with through HTML5, but it's not working properly in the mobiles actually. So,
okay.
No, I'm sorry.
So prefer from desktop. Okay.
Yeah, we are working on that, and actually at the end of the year we hope to have a project, an Android application and an iPhone application actually.
Yes, I understand that in a small screen you have to spot the cells, and it can be difficult.
Yeah, because I mean we'll need to have a really high resolution, and it's not possible, so they take a lot of space in the mobile phone. And some of them, you can use it, but the look and feel, the look and feel is not so good.
Yeah, okay.
Sometimes it not works. It depends on the mobile phone also. So, well.
Okay, thank you.
You're welcome. Here you have the application. So from here you can start contributing. But first we have a tutorial that I'm gonna skip now, but you should read it if you want to use it.
So we have to click on participate, right?
Yeah, and start contributing, I think. And yeah, this is the application. So here it appears the images from the cell culture, and that's got a. I don't know if you want to.
You can go ahead. You know more about the application than I do.
So the idea is to classify the images. On the left, you have samples, or always on the right, you always have the samples that you can compare the cells on the image on the left. I don't know if everyone. I skipped the
tutorial. Yes, I also skipped the tutorial. So I see now a photograph on the left and then some examples on the right.
Yeah, exactly.
Example of live cells and example of death cells.
Yeah, exactly. So on the right you always have the samples, and on the left the image to be analyzed. As you can see on the bar below the image, there are 4 steps. The first one to mark the alive and the death cells. The second one for mark the cells that are releasing their content. The third one for the mitochondrial distribution, and the last one is the remarks. So, in the first step, we have to mark all the cells. So it's pretty easy. Just click on the kind of mark you want to do. For instance, if we want to mark the alive cells, we make click on the heart, the first icon that says alive, and we can just click on on top of the cells, above the cells that we think they are alive. Okay, it's pretty easy.
So you can check if they're alive, death.
Yeah, for instance,
up to types of death.
Yeah, related with this, we have one question also in the chat. If you measure the accuracy of people identifying the cells, either apoptosis or necrosis, so what if I do it wrong? You know, what if I place the live cells whatever in the image?
First, when I obtain these images, I analyze them, so I know basically what's happening there. Also, we want to carry out in parallel an automated computing analysis of these images. Basically, a computing image analysis software. And then we will compare both. Also, each image is sent to several people, so we have some statistics on how reliable the analysis.
Yeah. So a lot of people check the same image. Yeah.
Several people are going to analyze the same image. If we see something that doesn't match because it has a high standard deviation, we will look at it more closely.
Okay.
Yes, every image it's sent to 25 different people. So we get from each image 25 different replicas that we can just run a statistical analysis.
Yeah, I understand. I had no that idea. Good.
Okay, and as Jose said, at the same time we are working on computer vision algorithms and machine learning to try to automatize the process. So at the end,
but if you do that, you don't need people anymore. So citizen science will not be needed.
But the point is that we need to train those algorithms because all those algorithms are already written, but the efficiency is not as good as it should be. So we are working we're working on the machine learning algorithms to improve those divisions. And to do that,
people will help to improve that algorithm and that algorithm to learn.
And the data set. What we need is a big, really huge data set to train the machine learning. That is what we are getting with all the people working on that. Now we get the analysis that they do, and also we at the same time we are creating the data set to train the computer.
Yeah.
And in such a way, we need the people to carry out that analysis. Yeah. So just pretty easy to mark, and as you can see, it's something that everyone can do. And in the field of education, it's really nice because they see a real cell, and it's not they really have the images that the Cote has got from the microscope. So they are really doing science, and they are doing the same classification that Jose has to do.
Yeah.
And they really get
this can be done by anyone, by pupils, of any age.
And right now it's in Aragon, or region in Spain. There are 35 schools that are working on that. In Madrid, there are several. In Portugal, I think there are also like 20 schools that are working with it. And in Japan, they started now in January. And Austria and Brazil are to come also. They are talking with the schools to start in a couple of months. So yeah, it's this application particularly fits pretty well in the education field because all the biology, it's easy to understand, and they can get a really strong background on what is apoptosis, necrosis, and real cells. So yeah, and there are also for the teachers, we prepare a didactic unit that they can use with the pupils to get some background for the application, and well, that's all about this application. Right now, I would like to share with you the video of the application that it spans a bit. I mean, it says more or less what Kotre told us before, but here you have it. So we can just leave the video for.
All living beings are made out of cells. Plants and animals contain many different cells. There are also unicellular organisms made out of a single cell, as protozoa, yeast or bacteria. All cells are originated by division of other cells, except the first cell that appeared millions of years ago at the origin of life. Pluricellular organisms usually reproduce sexually. After fecundation of an oocyte by a sperm cell, the new cell
what happened?
All living beings are made out of cells. Plants and animals contain many different cells. There are also unicellular organisms made out of a single cell, as protozoa, yeast or bacteria. All cells are originated by division of other cells, except the first cell that appeared millions of years ago at the origin of life. Pluricellular organisms usually reproduce sexually. After fecundation of an oocyte by a sperm cell, The new cell, known as zygote, begins to divide, forming an aggregate of many identical cells. During embryo development, these cells differentiate into several cell types, giving shape to the embryo through morphogenetic movements. A human being contains around 200 cell types. Each cell type occupies a defined place within the organism, specializing in specific tasks. Cells within a living being are precisely coordinated to allow survival of the organism, forming tissues and organs. Some cells, as neurons, are not usually replaced by others when they die. Nevertheless, inside most tissues and organs, cells age, die, and are replaced by others through the process of cell division. Our organs function correctly due to the precise coordination of their cells. Each cell is a complex living being that contains small organs known as organelles, as mitochondria, or the endoplasmic reticulum. Proteins are essential molecules for life. Many of them are real microscopic machines specialized in certain tasks. Some proteins function by copying fragments of DNA that contain genetic information.
What?
All living beings.
Maybe we can just we will provide you the link so you can take a look later. I don't know why it stopped again. And what more or less this is what we do in the institute. The main research lines and about us, the computation part and the citizen science part. This is our main line, and well, later we will provide you all the links and the links to the green paper and all the didactic unit, the videos, and all the links that we have shown. And no, thank you very much for your attention. If you have,
thank you. It was very interesting, and also for everybody, thank you for coming. We will do another Meet the Scientists in the future. And also, if you have any question, you can get in contact with Gina or Evita, and we will translate, give the questions to these researchers. Well, Ivo Jokin from Bulgaria says that as head of the Municipal Center for Extracurricular Activities and Science teacher, he will try to organize young people special training and presentation of this project. So maybe you can get in contact with new schools or new people or new teachers just to collaborate in your citizen science.
Yeah, sure.
Okay, so thank you.
That's I will provide also our contact, and we'll be really keen to collaborate with you and whatever you need, or just drop a mail. And
yes, I think also that the students recognizing cells and working with real photographs of cells can motivate them for their future works and to decide what they want to do in the future. They can see that that is a real thing, not a thing that also is on the movies.
Yeah, I mean, the idea is that they really get involved in the process of science. They see that it's not something so far, but it's something that everyone can collaborate. Yeah. It's a like of some
they can feel like part of it. Yeah.
And for the students, I think it's pretty motivational, and also they use the things that they are used to use, like all the technology, all the PCs and computers. That is not they get out of the books, and they use what they are really used to have in their hands, the mobile phones, the computers, and it can also foster the technological careers, and it's pretty interesting.
Yeah, there is also another question in the chat about the video. Indeed, we will send it. It's in YouTube, so you will have the video and you will be able to see it. If you have any question about the video, you can get in contact with us.
If you just look for Scientists in YouTube, you will find it.
Okay, we will send a report.
We'll send down the link. So
yes, I will send a report with everything, with all the links, the BIFI page, web pages, everything that we have shared, also the presentation that is on Slicer, and the YouTube videos. And also this recording, the recording of this session.
There are also some pictures of the laboratories on our web page, so you can go through it and see exactly different equipment that we have, the microscope, many different things.
Okay, thank you. Thank you very much.
You're welcome.
You're welcome.
Bye.
Bye.
Bye.